Good morning, everyone.
Good morning, ma'am.
Good morning, ma'am.
How are you all?
Fine.
आज ठीक है पंकज?
हाँ मैम, बट थोड़ा सा वो ज़्यादा हो गया था, बट देन मैंने दवा दवा लिया।
अच्छा, फीवरिश लग रहा है?
नहीं, फीवरिश नहीं लगा, मैंने वो पैरासीटामोल ले लिया।
अच्छा, और बाकी
हाँ, बाकी मैंने रेमेडीज ट्राई किया।
अच्छा, बट कंटिन्यू टेकिंग स्टीम, हाँ?
यस मैम।
हाउ आर यू उर्वशी?
Fine, ma'am.
गांव जाके आ गई? गांव जाके आ गई क्या?
गांव में हूँ।
अच्छा, गांव में ही है, ओके। Naina has joined. Somnath is there. Somnath has joined quite a long time back. Good morning. कसा आहेस तू सोमनाथ? मस्त ना? बस मजेत. ह्यांचं प्रवीण सरांचं लेक्चर आहे ना? विसरले की प्रवीण सर, नंतर निशा मॅम आणि नंतर कोण? नम्रता. अच्छा, सदा has Gayatri joined? No. Yes.
Okay, yeah. Good morning.
Good morning,
sir.
हाँ,
Good morning, sir.
कुछ पढ़े आप लोग? Any doubts till now? मैं लास्ट आपका मेरा मंडे लेक्चर हुआ ना, उसके बाद में मैं डाला नहीं, बट टुडे वंस वी कंप्लीट आई विल पुट अप द पीडीएफ या जो भी है, तो मैं एक्स्ट्रा रीडिंग मटेरियल भी डालूंगा. ओके, बिकॉज नेक्स्ट वीक आपका क्लास टेस्ट रहेगा एमसीक्यू टाइप. ओके, साथ में मैं आपको एक असाइनमेंट क्वेश्चन भी देने वाला हूँ. तो जिन्होंने पहले क्वेश्चन सबमिट नहीं किया है वो प्लीज इस वीक सबमिट कीजिए. ओके, सो दैट आपका क्लास टेस्ट के लिए भी उतना रीडिंग हो जाएगा. आगे हो सकता है नेक्स्ट मंथ ऑल्सो वी मे हैव इवेल्युएटिव टेस्ट ले सकते हैं, डिस्क्रिप्टिव टाइप, ठीक है. सो रीड. ओके, विल नाउ स्टार्ट. ये हमने कंप्लीट किया है नाउ. Okay, this all we have completed. उसका revision लिया है. This is what we had started with. Okay, and 50% पहुंचे थे. Okay, we had talked about purification. As I told you, purification goes hand in hand with your isolation part. Purification can be divided into two. That is a preliminary and secondary. Preliminary again, it goes again together with isolation. जिससे हमने self fractionation बोला था. Self fractionation is nothing that goes with along with centrifugation. Okay, जिसका मैंने एक slide दिखाया था पिछली बार. Right, this is what is you talk about self fractionation. जहाँ पे you mince या homogenize, and then later on you subject it to centrifugation, differential centrifugation, and you try to isolate the fraction. Okay. Next, we have talked about use of enzymes. Okay, sometimes enzymes may be used to lyse and extract the enzymes. Okay, then you talk about solvents are used, or sometimes precipitation can be carried out by using maybe ammonium sulfate or so. Okay, or sometimes enzymes, temperature may be required, may be employed, wherein enzyme of interest may be stable and other enzymes, right, which has been extracted may get precipitated or denatured. So that was the preliminary part. Next, to talk about the secondary, we have talked about sometimes the enzyme extract, right, may contain certain colored substances or pigments. And for this purification process, sometimes these pigments or the coloring agents has to be isolated. So some adsorbent gels may be employed. Now, when you talk about adsorbent gels used, it is not like, it is something similar to you talk about the chromatography, adsorption chromatographic technique type, where adsorbent gels are used and the extract is what to say employed, or it is put onto the column, and the pigments get adsorbed and the enzymes of interest, right, can be extracted, or they become purified from the pigmented. Next technique, right, is electrophoresis. As you can see on the slide, electrophoresis and chromatographic techniques, right, these are employed for purification. While the last three जो है lyophilization, ultrafiltration and crystallization, these are not basically, you can say, techniques, but rather they are employed, they are the steps, right, employed after the purification. That means electrophoresis, chromatography, जो भी है, these are employed for purification. And once you have the purified, sometimes you may have to, क्या बोलेंगे, it contains some solvents, right, and this has to be removed to get our enzymes in highly purified form. So lyophilization, ultrafiltration, crystallization are the steps involved, rather than being type of secondary purification. Now, last time we have talked about electrophoresis. Now, what is electrophoresis? Right, electrophoresis, last time I talked, right, the basic principle of electrophoresis is on what you talk about separation of the molecules based upon charge to mass ratio. Charge to mass ratio. Now here the molecule, now when you talk about electrophoresis, electrophoresis can be grouped or classified into two. One is what is called as the zone electrophoresis, and the other one is called as a moving boundary electrophoresis. Now, zone electrophoresis is a type where you require where you use a supporting medium. A supporting medium is used on which the sample is loaded, an electric field is passed, and the molecules are separated. While when you talk about a moving boundary electrophoresis is one where a supporting medium is not involved, and that is why the name moving boundary, right? There is no supporting medium. So rather I would say more or less the medium which is used is liquid, and the molecules are you can say injected, or they are loaded, I would say, and electric current being passed, and the molecules are separated. So as you can see on the slide, right, electrophoresis, I have shown three: zone electrophoresis, isotachophoresis, and isoelectric focusing electrophoresis. Now, out of the three, right, zone electrophoresis, right, is a, as I told you, is a separate type of electrophoresis where a supporting medium is used. Now, zone electrophoresis, there are different type of zone electrophoresis, right? Maybe paper electrophoresis or the commonly employed gel electrophoresis, which we use, right, is also a type of zone electrophoresis. Isotachophoresis and isoelectric focusing, they are mostly come under what you call as a moving boundary electrophoresis. Now, zone electrophoresis, as I told you, right, it involves a supporting medium, right, which can be starch gel or maybe the most commonly employed acrylamide gel, that is a polyacrylamide gel, or maybe agarose gel, right? And you use a buffer, the electrolytes, electrodes are there, you have the anode and the cathode. My sample, right, is loaded and a current is passed. Now, see here in case of zone electrophoresis, the charge is maintained uniform. The pH is maintained uniform. So what happens is that the separation occurs based upon the size of the molecule. And you are aware that smaller the size, it will run ahead, or faster will be the mobility and it will run ahead, while larger the molecule, slower will be the mobility and it will lag behind. So on separation, right, here you use a tracker die. You can use a tracker die to see where till where the separation has taken place, and once it has reached the end, you stop the electric current, and you can employ the what do you call a staining technique. Particular staining technique can be employed where the you get colored bands, and once the bands are obtained, you can cut the. Now when you talk about enzyme, these samples are loaded. You can load in more than one wells. So in one well you have the entire stretch of the band. So a longitudinal cut can be made, right? And you can analyze how many bands are there and where are they. The what you call as a RF, you can find out. Alternate the remaining part, you can cut what is it perpendicular to the mobility, to the direction of the mobility. So you get different parts of the gel at different level, and each section can be analyzed or the fraction isolated and can be analyzed. Okay, and in this way, right, you can isolate your enzyme of interest in purified form. That means when I talk about horizontal stretch पूरा है, तो you can locate कि आपका enzyme कहाँ पे है, and बाकी के part से आप जब उसको perpendicularly section cut करते हैं, तो जहाँ पे आपका enzyme of interest है, वो आप section ले सकते हैं, and then you can isolate that enzyme of interest from the gel and further subject it to purification. So that is your zone electrophoresis. Okay, isotachophoresis, right, is a type of, as I told you, moving boundary electrophoresis. Right now, moving boundary electrophoresis में क्या है? As I told you, mostly the supporting medium is not used. Supporting medium is not used. Now, what is done here? Last time I told you here, I showed you a diagram, right? Where mostly, right, isotachophoresis is carried out horizontally, but then you have vertical type of isotachophoresis also. Okay, in short, it's called a IT, isotachophoresis, and isoelectric phoresis is IEC. Isoelectric IEF. Okay, now as you can see on the diagram, क्या है? You have an anode and cathode, right? As I told you, right? You have a leading electrolyte, right, and a terminating electrolyte. Similarly, right, in isotachophoresis में क्या है? Here the separation, right, it is derived from a Greek word. It is derived from a Greek word. क्या है? Iso means same. Iso means same, and tacho means speed. So here the molecules are separated, right, based upon the speed. Those molecules having the same speed will go together. So now what is done here, right, in isotachophoresis, mostly smaller molecules are separated. तो क्या करते हैं इसमें, right, I use a क्या बोलेंगे, leading anion या leading cation use होता है. Similarly, termin, agar if I suppose my protein of interest, right, is क्या बोलेंगे, is an anion. ओके, तो क्या करता हूं मैं, अगर एनायनिक है, एनायनिक मतलब क्या है, इट विल मूव टुवर्ड्स एनोड. हेलो,
You're audible.
हाँ ऑडिबल.
Yes, yes.
नहीं बच्चों को पूछ रहा हूँ, तो समझ में आया क्या?
Yes, sir.
हाँ, सो इसमें क्या करते हैं, सबको माय प्रोटीन ऑफ इंटरेस्ट, नाउ इट कैन बी पॉजिटिवली चार्ज, नेगेटिवली चार्ज हो सकता है, राइट. सो दैट इज ऑन दैट बेसिस, एज आई टोल्ड यू, इट कैन बी एनायनिक और कैटायनिक. ओके, जो भी अगर एनायनिक अगर है, राइट, तो क्या होता है, आई यूज लीडिंग एनायन यूज करता हूँ और एक टर्मिनेटिंग एनायन यूज करता हूँ. Similarly, it is cationic है, then I use a leading cation and I use a terminating cation. Now अभी ये क्या चीज है? Now usually when I use, अब समझो anionic protein है, right? अभी anionic protein है मतलब क्या होगा? It will move towards क्या होगा? Anode. It is moving towards anode. तो मेरा क्या रहेगा? My protein is a negative charge. बराबर है? Okay, so what I do is, I apart from the sample, तो मेरा setup तैयार होने के बाद, right, I use a leading anion which is a very small molecule, okay, and naturally which will run ahead. जैसे मैंने कहा speed पे जाता है, current when it is fast, तो क्या होगा, right, smaller molecules will run ahead. So a leading anion डालते हैं, right, and that will be towards the anode. When I say towards the anode, मतलब ये नहीं कि एनोड के पास, ओके, जहाँ पे मेरा लोडिंग रहेगा एक साइड में, तो आगे के साइड में मतलब टुवर्ड्स द एनोड साइड बोल सकते हैं, तो वहां पे मेरा लीडिंग एनायन रहेगा. एंड सिमिलरली कैटायन की तरफ मतलब पीछे के साइड में, राइट, दैट विल बी द टर्मिनेटिंग एनायन, ओके, लार्जर मॉलिक्यूल. और ये दोनों को बीच में, राइट, मेरा सैंपल रहेगा जो मेरे को सेपरेट करना. ये सेटअप होने के बाद में करंट पास करते हैं एंड नाउ इट विल स्टार्ट मूविंग. सो लीडिंग एनायन क्या रहेगा, वो आगे जाएगा, माय टर्मिनेटिंग एनायन पीछे रहेगा, और बीच में मेरा सैंपल रहेगा. और सैंपल मॉलिक्यूल क्या हो जाएगा, डिपेंडिंग अपॉन द साइज, राइट, दे विल सेपरेट आउट. जो मॉलिक्यूल सेम साइज के हैं वो साथ में जाएंगे, तो दे विल फॉर्म द बैंड. तो एक पर्टिकुलर टाइम होने के बाद क्या दिखेगा, राइट, दिखेगा नहीं, और क्या होने वाला है, इज दैट द मेरा जो एंजाइम मिक्सचर जो है, जो भी है, राइट, दे हैव सेपरेटेड आउट एंड आई विल गेट जोन्स. मैंने से जोन मतलब क्या, बैंड जैसा दिखेगा. दैट ईच बैंड या जोन में क्या रहेगा, दे विल हैव मॉलिक्यूल्स ऑफ द सेम साइज और दे आर व्हिच आर मूविंग विद द सेम स्पीड. ओके, राइट. So this is how the isotachophoresis is used, right? अब isotachophoresis जो है, इसमें भी बहुत सारे modifications आए. You have जैसे मैंने कहा सपोर्टिंग मीडियम नहीं यूज करते, लिक्विड है, but then I have gel isotachophoresis also, wherein a supporting medium is used or something like a gel, and the same process, मतलब something similar to like क्या बोलेंगे, gel electrophoresis आ रहा है, but then here I'm using a terminating and a leading anion. Similarly, if my enzyme of interest, right, is a cation, so क्या करूँगा, I use a leading cation and a leading anion. Same thing, I will load at one side, leading cation will be ahead, a terminating cation will be behind, और ये दोनों के बीच में, right, my extract load किया जाएगा, current will be passed, and the same thing. Okay, this is what is called as isotachophoresis. Now, all these, as I told in the beginning, chromatography, electrophoresis, these are highly sensitive techniques, right? Wherein very low amount of sample can be employed as well, but then at the same time you will get a higher resolution, and that means higher type of purity or purification. Okay, so this is isotachophoresis. Now, next one you talk about is isoelectric focus. Now, isoelectric focusing, जो IEF, right? That is also a type of moving boundary electrophoresis. अब इसमें भी modification है, जेल और use करते हैं, but again the principle here जो isoelectric focusing में क्या है? Now, in case of isotachophoresis में क्या था? There was a uniform, there was no zonation of the buffer. Okay, but pH was uniform throughout. But in case of isoelectric focusing क्या होता है? I create a zonation, right, of pH. Buffering जो है, there will be a different grades of what is say buffer, mixture of buffers I would say, but mixture in the sense वो वैसे mix नहीं करते हैं, right? My buffers are added and I get a gradation from one end to the other end. I can get a different pH range. Okay. So, and in this, right, I load the sample. But before loading the sample, right, क्या करता हूँ मैं? As you can see on the slide, a pH gradient is set up between the electrode. तो मैं एक साइड में एसिड डालूंगा, एक साइड में बेस डालूंगा, and slowly they will diffuse. So क्या हो जाएगा? I will get a gradation of the pH. Similarly, in this case, I can use in isoelectric focusing, right? I use certain क्या बोलेंगे ampholytes. Certain ampholytes. Now, what are ampholytes? Ampholytes are nothing but they are mixture of synthetic and natural amino acids, right? ये मैं load करता हूँ, right? And a current is passed. Once the current is passed, क्या अगर इधर now I have a pH zonation, and क्या होता है now proteins, right? As you know, they are pH specific, right? So the proteins may be charged, may be uncharged, but at a particular pH, right? क्या हो जाता है? The charge on the protein may be nullified. That means neutral हो जाता है. So that pH at which the protein gets neutral is called as the isoelectric point. Okay, so isoelectric focusing में क्या किया जाता है? Is that I see that the protein at its isoelectric point gets neutralized or gets precipitated. Right, so as I told you, I create a gradation, then I use a ampholytes, right, that is mixture of natural and synthetic amino acids, उसको load करते हैं, then I run the electrophoresis, and at the isoelectric point, each amino acid of the mixture जो है ampholytic mixture वो precipitate हो जाए, that means they will become neutral, and उसके बाद में आप कितना भी current pass करो आगे move करने वाला नहीं है, at the isoelectric point पे पहुंचने के बाद, right, वो move करने वाला नहीं है, वहीं पे वो स्टेबल हो जाएगा. सो आई गेट डिफरेंट क्या बोलेंगे एट दिस आइसोइलेक्ट्रिक पॉइंट पे दिस सिंथेटिक एंड जो नेचुरल अमाइनो एसिड जो मैंने लोड किए मिक्सचर वो अपने अपने बैंड्स फॉर्म करेंगे. नाउ ये होने के बाद में देन व्हाट आई डू इज आई इंट्रोड्यूस माय सैंपल. आई इंट्रोड्यूस माय सैंपल मिक्सचर ऑफ एंजाइम जो भी है, एंड नाउ द करंट इज पास्ड, एंड ये करंट पास जब करते हैं तो क्या हो जाएगा? अगेन इफ मिक्सचर जो है एट इट्स इलेक्ट्रिक पॉइंट ईच अपना एंजाइम जो है वो अपना स्टेबलाइज हो जाएगा, ईच एंजाइम जो है स्टेबलाइज हो जाएगा, राइट, और यू से इट बिकम्स न्यूट्रल. नाउ दिस पर्टिकुलर प्रोसेस जो है इट डिपेंड्स, इट मे टेक सम few hours or maybe it may take a day or more, okay, to stabilize or these molecules to reach their isoelectric point. जो भी है, right? Now what I will get is I will get bands, and as I told you these are highly sensitive because इसमें क्या हो जाता है pH वेरिएशन होने के कारण में, right? आप अगर सपोज आइसोइलेक्ट्रिक पॉइंट एक का किसी का 5.7 है, दूसरे का 5.8 रहेगा या 5.723 अगर भी रहेगा तो भी ये सेपरेट होके सेपरेट बैंड्स बन सकते हैं, शार्प बैंड्स बन सकते हैं, right? So that is how it is specific. वंस ये होने के बाद में जैसे मैंने कहा, right, you can carry out various staining technique and find out where my enzyme is of interest is located. Okay, here I can use specific substrate डाल के enzyme substrate reaction डाल के a coloring thing can be developed and I can locate where my particular enzyme is at, and then फिर वो उस band को जो है और उस area को isolate करके, right, I can purify my enzymes. Is it clear? So this is what is called as isoelectric focusing. So isoelectric focusing is also a type of moving boundary electrophoresis, right? और उसमें भी बहुत सारे प्रकार हैं, but then क्या होगा इधर you get sharp bands, okay? That means थोड़ा सा भी पॉइंट पीएच का पॉइंट का इधर उधर भी अगर फर्क रहेगा तो भी एंजाइम्स क्या हो जाएगा, वो अपने आइसोइलेक्ट्रिक पॉइंट पे सेपरेशन होंगे. Is it clear? So they are highly specific, right? So this is what is employed for purification. So what we have seen is that here, so we have talked about zone electrophoresis, isoelectrophoresis, and isoelectric focus. Here we'll be not talking about the instrumentation part and all, because since we are talking purification, we just have to know the principle and how it is employed. Okay, so this is how the electrophoresis technique can be used for purification. Another technique which can be used for purification are the chromatographic technique. Now here I won't say which one is highly specific, chromatographic hai ya it all depends upon pluses minus. Okay, but yes, it depends upon my enzyme जो है. So by trial and error, right? Sometimes, as I told you, one or more than one type of technique may have to be employed for purification. Now chromatographic technique, as you are aware, right? Again, it is a highly sensitive technique. Uh, and here basically यहाँ पे charge नहीं आते, you don't pass a electric field, but rather क्या हो जाता है, here the molecules are separated based upon क्या बोलेंगे उसका properties, right? Whether polar है, non polar है, क्या है, that can be employed. Now chromatic हम color बोलते हैं, but not necessarily always it is color. But then what are the case, here it can be separated based upon size आता है यहाँ पे, साइज के बेसिस पे सेपरेट होता है, पर अगेन इसका जो है प्रॉपर्टी पे भी डिपेंड होता है. ऑन दैट बेसिस द पर्टिकुलर मिक्सचर इज सेपरेट. नाउ क्रोमेटोग्राफी टेक्निक जो है, बहुत सारे क्रोमेटोग्राफिक टेक्निक है, राइट? इसमें आप साइड पे देख रहे हैं, राइट? वन इज व्हाट इज कॉल्ड एज आयन एक्सचेंज. कैटायन दिखा है, बट आयन एक्सचेंज क्रोमेटोग्राफी कैन बी यूज्ड. लास्ट टाइम वी हैड टॉक्ड अबाउट समथिंग ऑफ दिस, राइट? आयन एक्सचेंज क्रोमेटोग्राफी में क्या है, here, right, my molecules are charged. Okay, अभी अलग अलग chromatography के some properties are employed, and उस basis पे आप size तो आता है, but at the same time it depends upon some properties which are employed, and on that basis you have different type of chromatography. Now जैसे मैंने कहा adsorbent gels जो पहले बताया, right, that can be an adsorption type of chromatography. Right now here when you talk about ion exchange chromatography, this also can be a type of, you can say, adsorption type. Now here क्या हो जाता है is ion exchange chromatography. As I told you, I have a column, and in this column, right, I have what you call as ion exchangers. Okay, so I have to prepare a column, I have to pack the column with ion exchangers, and most of the time these ion exchangers are gels. Okay, gels, or they may be what you call as resins, right? Now different type of ion exchangers are used, or depending upon, either they can be cation ion exchangers or they may be anion ion exchangers. Okay, last time I told you the ion exchanger जो भी cation या anion exchanger exchange chromatography is named after the molecule which has to be isolated. अगर मेरा मॉलिक्यूल जो सेपरेट होना है वो अगर कैटायन है, देन इट इज कॉल्ड एज कैटायन एक्सचेंज क्रोमेटोग्राफी. अगर मेरा मॉलिक्यूल सेपरेट होना है वो एनायन है, देन इट इज कॉल्ड एज एनायन एक्सचेंज क्रोमेटोग्राफी. सिंपल चीज है, एनोड इज पॉजिटिव चार्ज, सो मेरा एनायन विल बी नेगेटिव. if I am talking about anion exchange chromatography, then मेरा सैंपल जो सेपरेट होना है वो एनायन है. एनायन मतलब इट इज नेगेटिव चार्ज. तो अगर नेगेटिव चार्ज है तो माय आयन एक्सचेंजर जो है वो पॉजिटिव रहना है. सो इन शॉर्ट सिंपल चीज है, एनायनिक एक्सचेंज क्या बोलेंगे क्रोमेटोग्राफी कहा तो माय आयन एक्सचेंजर जो है या द रेजिन व्हिच आई एम यूज इज पॉजिटिव चार्ज, and the molecule to be separated is negative, that is anion. Okay, similarly if I talk about cation exchange chromatography, same way, my molecule cathode is negative, so my cations are positive, so the resin which I am used will be what you call as the negative charge. Okay, so cation exchange क्रोमेटोग्राफी में क्या होगा, मेरा जेल जो यूज कर रहा हूँ, द एक्सचेंजर, आयन एक्सचेंजर जो यूज कर रहा हूँ, तो दैट विल बी अ क्या बोलेंगे, दैट विल बी नेगेटिव चार्ज, दैट विल बी नेगेटिव चार्ज, एंड जो मेरा मॉलिक्यूल सेपरेट होना है मीडियम में जो है वो पॉजिटिव चार्ज है, दैट इज कैटायन. ओके, नाउ एज आई टोल्ड यू, द आयन एक्सचेंजर्स जो यूज होते हैं, दे कैन बी नेचुरल और दे कैन बी आर्टिफिशियल, और दैट व्हाट यू कॉल एज सिंथेटिक. जो भी है दैट डिफरेंट, इसको आयन एक्सचेंजर जो मैंने कहा पैकिंग मटेरियल, दे आर आल्सो कॉल्ड एज मैट्रिक्स. And again it depends upon this cation exchangers or anion exchangers. They can be strong cation exchangers, similarly they can be strong anionic exchangers, similarly exchangers can be weak, that means they can be weak cationic exchangers or weak anionic exchangers. Sometimes when you talk about exchangers, right, they are modified, जैसे मैंने पिछली बार कहा, right, ion exchangers used. तो क्या यूज करते हैं, is either you use a DAE, right, or I can use what is called as CN cellulose, that is carboxymethyl, right. well, mostly when you talk cellulose बोलते हैं, so cellulose को फिर modify करते हैं, और I have carboxymethyl, उसको फिर I attach to cellulose and I get a CNC, that is carboxymethyl cellulose. Similarly I have what you call as DEAE, right. DEAE is nothing but diethyl aminoethyl, right, that is DEAE. इसको cellulose के साथ जोड़ते हैं, तो I get a DEAE cellulose, right. So DEAE cellulose is mostly used as an anion exchanger, okay, while CN cellulose is mostly used as a cation exchanger. क्या करते हैं इसमें, right, इसमें is nothing but I have column. Last time I told you I pack the column with this gel, okay. Taking care, now it has to be put into a buffer में अच्छे से stir करके उसको activate करते हैं, and then I carry out the packing of the column, taking care that there is no air gap, and the process has to be done very slowly ताकि ये gel sediment होके compact हो, okay. जो भी है, once my column is ready and then I put my sample, and sample को load करने के बाद में, right, slowly I start the process. जैसे मैंने कहा column है तो like a burette type, so I open the tap and slowly I adjust the flow, and धीरे धीरे जैसा वो buffer move करेगा तो my sample जो ऊपर top पे load किया है it will pass through the column, okay. Sufficient पहले load करने के बाद भी रोका जाता है, right, so that it can diffuse, and then slowly it is run. तो जैसे solvent move करेगा, right, my sample will also move across the gel and it gets time for to get exchanged. Or when I talk about exchange, मतलब क्या हो जाएगा, it will bind to the ion exchanger, right, और बाकी के जो unbinded है वो move out हो जाएंगे, right. And once this process is done, I continuously मैं क्या करता हूँ, buffer add करता हूँ, right. I note the time also, that is here in this case it is called as the retention time, okay, right. And I collect the fractions, and fractions को further में आगे analyze कर सकता हूँ कि किसमें है आया है, जो भी है, with particular note the volume of the buffer also being used, right. And then क्या करता हूँ, in this case I have to separate, मेरा जो अगर समझो enzyme molecule जो है वो bind हुआ है to the particular resin or the ion exchanger, then I have to carry out a process what is called as elution. Now elution is a technique जहाँ पे मैं I use a buffer of a particular strength, जहाँ पे जो binding हुआ है मेरा enzyme या protein molecule with the gel, वो break होना है, so that the enzyme or the protein of interest is get separated and comes into the solution, right, and then it can be removed or eluted out, okay. So this is what is done in cation exchanger. So here again it is a highly specific, okay. So if I know, right, protein उसका charge क्या है, तो accordingly I can carry out, or it can be by trial and error, okay. So जो भी है cationic anionic exchanger use करके, now again as I told you they are highly specific depending upon the charge or affinity, right. Affinity भी आता है towards the particular gel molecule, right, and separate किया जाता है. तो here क्या हो जाता है, my fraction जो मिला है, right, they are in highly purified form, right. Most of the impurities या most of the mixture जो है वो separate. So this is what is called as the ion exchange chromatograph. Another technique, as you can see on the slide, is gel filtration. Now, gel filtration, as I told you, it is also known by different names. Maybe it is also known by size exclusion chromatography or molecular sieve chromatography. Here also same thing. As the name suggests, gel filtration है. यहाँ पे भी beads use करते हैं, right? Bead use कर सकते हैं. They may be cellulose bead, agarose bead, जो भी है, it depends. But फर्क इतना है, ऊपर ion exchanger में क्या these beads पे charge, right? So I use and that is why they are known as ion exchangers. In case of gel filtration, I use the beads, right? But now they are not charged. But rather क्या होता है, इधर beads they are of different, they have to be of different size and they have to be of different porosity, right? तो इधर क्या हो जाता है, जब मेरा जो beads use किया है, they are porous and depend upon the pores उस पे जो है, छोटा है या बड़ा है. This is what, and that is why the name molecular sieve करता है. So जब मैं sample load करता हूँ, right, जो molecules एक pore size से छोटे हैं, right, वो क्या करेंगे, वो जेल के अंदर जाएंगे. जेल molecules जो है, bead जो है, उसके अंदर जाएंगे. They will be trapped in that. और जो बड़े वाले हैं वो अंदर जा नहीं सकते because the pores are smaller, right? तो ये क्या रहेंगे, ये जेल beads के बीच में रहेंगे, और you can say they are beads के बाहर है. Right, so जब मैं इल्यूशन टेक्निक करता हूँ तो क्या हो जाएगा, these molecules which have not been trapped inside the gel, they are the first one to move out जब मैं इल्यूशन करता हूँ. Okay, right, and my molecules of interest जो है, now it depends upon either वो बाहर रहेगा या अंदर रहेगा, जो भी है, but then separation हो गया depending upon the size, right? And then further मैं क्या करता हूँ, I carry out the elution technique, जहाँ से जो मॉलिक्यूल्स मेरे बीड के अंदर जो है उसको भी मैं बाहर निकालने की कोशिश करता हूँ. clear, right? I can change the what is it, buffer या eluting solvent, जिससे क्या हो जाएगा, affinity change हो, right? For pores का जो है ना, pore as such change नहीं होता, that will all depend upon the concentration, but still certain उसका जो flexibility of the pore जो है modify होता है. So my molecules जो अंदर trap हुए हैं वो बाहर आ सकते हैं, but that will be after. तो पहले जो ट्रैप हुए नहीं है वो पहले निकलेंगे, और जो ट्रैप हुए हैं, राइट, जो पोर से छोटे थे और अंदर गए थे, वो बाद में बाहर निकले. Okay, so this is how gel filtration is employed for separation technique. Okay, now then next, as you can see, right, is called as affinity chromatography. Now as the name suggests, affinity chromatography, यहाँ पे भी gel bead use होता है. But यहाँ पे क्या आता है इधर, right, in case of affinity chromatography, right, I use what is called as क्या ligand. Certain ligands are used. Okay, when you talk about cation या anion exchange, ion exchanger में क्या होता है, these beads are activated, charge लाता है इस पे, surface पे charge है. Okay, adsorption type है, but यहाँ पे charge है. Okay, जेल में चार्ज और कुछ नहीं, वो अंदर जाता है. So it's something like absorption type बोल सकते हैं. Absorption भी नहीं exactly, but then वो अंदर ट्रैप हुआ जाता है. In case of affinity क्या, affinity it's like a adsorption, surface पे attachment है, but then attachment कैसे किया जाता है, पहले तो मेरा जो जेल है उसको मैं रिएक्ट करता हूँ certain ligand से. Ligand मतलब certain molecule, right, जिसके कारण मेरा जेल और ligand, right, they are usually they may be non-protein moiety, right, and a binding is brought about between the gel and the ligand. Okay, in short I can say मैं activate कर रहा हूँ मेरा जेल को. In case of ion exchanger में activate करता हूँ, charge लाता था मैं surface पे. यहाँ पे मैं charge नहीं, यहाँ पे क्या करता हूँ मैं, I bind certain functional group बोल सकता हूँ, right, which are known as ligands. So once these are formed, right, ligand जुड़ गए मेरे जेल को, now my enzyme of interest is loaded, और mixture जो है load किया जाता है. अभी क्वेश्चन आता है कि ligands कौन से यूज करने. ligand वो यूज करते हैं जो मेरे एंजाइम जो ऑफ इंटरेस्ट जो है उसको बाइंड करेगा. सो मेरा एंजाइम ऑफ इंटरेस्ट का सर्टेन प्रॉपर्टीज अगर मुझे पता है, सो अकॉर्डिंगली क्या है, आई कैन यूज ligands एंड आई कैन क्रिएट अ जेल, राइट, बीड्स जिसको मेरे ligands, ओके, अटैच है, एंड दिस ligands क्या करेगा, दे विल स्पेसिफिकली बाइंड टू माय एंजाइम ऑफ इंटरेस्ट. ओके, so this is what is called a affinity chromatography. सो फर्स्ट आई हैव माय जेल को मैं क्या करता हूँ, इफ आई नो माय एंजाइम के प्रॉपर्टी मुझे पता है तो अकॉर्डिंगली मैं लिगैंड्स इनपुट करता हूँ, एंड देन आई मेक दिस लिगैंड्स बाइंड टू द जेल, द सपोर्टिंग मीडियम को मैं बाइंड करने देखता हूँ। तो वंस माय क्या होगा, द जेल्स आर एक्टिवेटेड विद द लिगैंड्स, लिगैंड जुड़े हुए हैं ऑन द सरफेस, देन माय मिक्सचर जो मैं लोड करता हूँ, एंड मिक्सचर जब मैं लोड करता हूँ along the buffer, क्या हो जाएगा, दिस लिगैंड विल स्पेसिफिकली बाइंड टू द एंजाइम ऑफ माय इंटरेस्ट, बाइंड करके रखेगा, बाकी को बाइंड नहीं करेगा, सो दे आर फ्री। सो क्या हो जाएगा, आई कैरी आउट द इल्यूशन प्रोसेस, इल्यूशन प्रोसेस में जो बाइंड हुए नहीं है वो अलग हो गए, राइट, और मेरा जो एंजाइम है वो लिगैंड के साथ जुड़ा हुआ है, तो सेपरेशन हो। नाउ मैं क्या करता हूँ, आई फर्दर मॉडिफाई द बफर, और मैं ऐसा डालता हूँ जिसके कारण मेरा लिगैंड और एंजाइम का जो बाइंडिंग है वो ब्रेक होगा, सो माय एंजाइम ऑफ इंटरेस्ट विल बी सेपरेटेड, राइट, एंड देन आई कैरी आउट द इल्यूशन, तो माय एंजाइम ऑफ इंटरेस्ट जो है, राइट, वो सेपरेट होगा। नाउ ओनली थिंग इज व्हेन यू टॉक अबाउट एफिनिटी क्रोमेटोग्राफी में क्या है, हियर सर्टेन हार्शर, जैसे मैंने कहा लिगैंड है, लिगैंड को बाइंड करते हैं, तो यूजुअली लिगैंड एंड बाइंडिंग जो है विद माय एंजाइम या प्रोटीन, राइट, दे आर मच टाइटर। तो अगर मुझे इल्यूट करना है, तो उसको सेपरेट करना है, तो क्या हो जाता है, समटाइम्स, राइट, माय कंडीशंस एम्प्लॉयड, जैसे मैंने कहा पीएच एंड ऑल, तो थोड़ा सा हार्शर हो सकता है, जिसके कारण, राइट, माय प्रोटीन और माय एंजाइम ऑफ इंटरेस्ट में गेट डीनेचर, तो ये होने का चांस है। ओके, बट द नेवरदलेस क्या है, बीइंग लिगैंड्स बीइंग स्पेसिफिक, क्या हो जाएगा, यू गेट अ स्पेसिफिक बाइंडिंग, राइट, एंड एक्चुअली द सैंपल जो मुझे मिलेगा एट द एंड विल बी हाईली प्यूरिफाइड फॉर्म। Okay, so this is what is called as affinity chromatograph. Next आता है एक modified of affinity chromatography is what is called as immunoaffinity chromatography. As the name says, जैसे मैं something like ELISA में बोल सकता हूँ, right? तो क्या करते हैं, इधर ligand ही है, ligand ही use करते हैं, right? But then the ligand used here, right, is an antibody to the enzyme or protein of interest. ये होता है। इन केस ऑफ एफिनिटी में क्या है, दे आर नॉट एंटीबॉडीज, राइट, दे आर दे कैन बी, क्या बोलेंगे, सिंथेटिक। हियर जो एंटीबॉडी यूज होता है, नाउ हियर एंटीबॉडीज एज अ नेम सजेस्ट, देन दे देमसेल्व्स आर प्रोटीन्स, द एंटीबॉडीज व्हेन आई से दे देमसेल्व्स आर प्रोटीन। इन एफिनिटी में जहाँ पे मैंने बोला लिगैंड, दे में नॉन प्रोटीन हो सकता है, बट इधर एफिनिटी में क्या है, लिगैंड्स आर देमसेल्व्स प्रोटीन। Okay, so these molecules are employed, and here क्या हो जाता है, antigen antibody, so that means my enzyme of interest जो है is basically functions as an antigen. Now when I talk about these antibodies which are used as ligands, actually वो पूरे protein molecule नहीं होता है, but they are basically such that कि वो मेरे जो protein of interest है, तो उसमें जो functional group के साथ binding हो, so there is a specific antigen antibody specificity binding जो है, तो फंक्शनल ग्रुप के साथ होता है। तो एंटीजन जो मेरा एंजाइम ऑफ इंटरेस्ट अगर जो है, उसके जो फंक्शनल ग्रुप है, उस फंक्शनल ग्रुप के साथ बाइंड होना। सो इधर आई व्हाट आई यूज इज, जो कहा मैंने, एंटीबॉडी टाइप यूज करते हैं, राइट, एंड द एंटीजन एंटीबॉडी रिएक्शन जो है, दैट प्रॉपर्टी इज एम्प्लॉयड हियर इन केस ऑफ इम्यूनो एफिनिटी क्रोमेटोग्राफी। ओके, एंड देन फर्दर सेम थिंग, राइट, बाइंडिंग हो गया, एंड देन फर्दर इल्यूशन हैज टू बी ऑकर्ड, बट इधर क्या हो जाता है, व्हेन आई कैरी आउट द इल्यूशन, मतलब क्या होगा, पहला जो बाइंडिंग हुआ नहीं है मॉलिक्यूल्स, राइट, वो निकल गए, मेरा एंजाइम ऑफ इंटरेस्ट वो बाइंड हुआ है लिगैंड के साथ, राइट, द एंटीबॉडी के साथ में। नाउ आई हैव टू ब्रेक दिस एंटीजन एंटीबॉडी, बट एंटीजन एंटीबॉडी ब्रेक करने के बजाय इधर क्या होता है, द लिगैंड को ही सेपरेट किया जाता है, राइट, आई ट्राई टू सेपरेट आउट द लिगैंड। ओके, तो लिगैंड एंजाइम जो है वो मॉलिक्यूल सेपरेट हो जाता है, दे बिकम फ्री, they come to the solution, and then elute होने के बाद में then I try to separate out the what is it, the ligand enzyme react what say molecule, उसमें से मैं enzyme को purify करने को देखता हूँ। Okay, so this is what is called as immuno what say affinity chromatograph. Okay, next आता है is called as HPLC, right, that is high performance liquid chromatograph, right, this is another technique. Chromatographic technique was a highly precision technique, okay, जहाँ पे now इसमें भी कॉलम यूज किया जाता है, पर यहाँ पे जो HPLC में कॉलम जो यूज किया जाता है इस अलग ही है, राइट, दे आर क्वाइट कॉस्टली, स्टेनलेस स्टील का कॉलम यूज किया जाता है, एंड इसके अंदर पैकेजिंग मटेरियल होता है, इसके अंदर पैकेजिंग मटेरियल। बाकी के इसमें क्या है, जो कॉलम यूज करते हैं दे कैन बी, इट डिपेंड्स अपॉन डिफरेंट, ग्लास के हो सकते हैं, प्लास्टिक के हो सकते हैं, जो भी है। बट इन केस ऑफ HPLC, राइट, एंड बेसिकली इट इज हॉरिजॉन्टल, द कॉलम्स आर प्लेस्ड हॉरिजॉन्टल इन द इंस्ट्रूमेंट, राइट, एंड द बॉडी जो है कॉलम का, इट इज मेड अप ऑफ स्टेनलेस स्टील, राइट, एंड अंदर मेरा कॉलम पैक किया जाता है, एंड दिस कॉलम्स आर वेरी वेरी नैरो, ओके। पर उसमें भी कॉलम्स में भी दो प्रकार के हैं, they are some are wider and some are narrower, so based up depending upon whether it is preparative जाना है या analytical technique जाना है। Preparative mostly the columns are wider, okay, चौड़े होते हैं, and in case of analytical the columns are narrower, because मेरे यहाँ पे more precision आता है, okay। जो भी है the columns, यहाँ पे एक जो column है वो बनाया बनाया मिलता है, because you cannot prepare the column because it is highly precision है, and each column is highly very very costly, okay। So and again in most of the cases if your sample जो loading है they are not purified to a certain extent, then my column can get contaminated, yeah, they may be spoiled, and this is not good, okay। So certain care has to be taken when you are using HPLC, right, जो भी है, right, HPLC जब use करते हैं तो as a name suggest high performance, sometimes it is also known as high pressure chromatography, right। इसमें भी हर फर्दर मॉडिफिकेशन आई कैन यू, जैसे मैंने कहा आयन एक्सचेंज है तो उसमें भी हाई परफॉर्मेंस आयन एक्सचेंज क्रोमेटोग्राफी है। वैसे इधर व्हेन यू टॉक अबाउट हाई परफॉर्मेंस, राइट, हाई प्रेशर, सो यहाँ पे प्रेशर यूज किया जाता है दिस HPLC, right। So again it's not like manual type, the sample has to be injected, so there is a syringe type mechanism है। पहला मेरे को मेरे इंस्ट्रूमेंट को क्या करते हैं, आई कैन से स्टैंडर्डाइज किया जाता है, एक्टिवेशन फॉर्म में होना चाहिए, रन किया जाता है विद द सॉल्वेंट एंड ऑल, ओके, पहले बिफोर द सैंपल इज लोडेड, सो दैट दे आर सैचुरेटेड, राइट, एंड उसके बाद में मेरा सैंपल जो है वो प्लेस किया जाता है, उसमें पर्टिकुलर प्लेस है जहाँ पे मैं सैंपल प्लेस कर सकता हूँ, एंड देन विद मैकेनाइज्ड फॉर्म, राइट, देयर इज अ सिरिंज टाइप, उसमें मैं कंप्यूटराइज्ड है पूरा, सो आई कैन पूरा डाल सकता हूँ कि कितना सैंपल लेना है, कितना सैंपल इंजेक्ट करना है, जो भी है, कितने टाइम रन करना है, ऑल दैट थिंग इज वर्ड से कंप्यूटराइज्ड, राइट। तो सैंपल फिर इंजेक्ट किया जाता है, राइट, into the what is called as the column, right, and then slowly क्या हो जाता है, जैसा मैंने कहा मेरा बफर जो है वो तो मूव करते रहता है, एंड देन स्लोली क्या हो जाएगा, द सैंपल लोडिंग होने के बाद में दैट विल आल्सो मूव अलोंग विद द बफर, राइट, एंड डिपेंडिंग अपॉन द एफिनिटी जो है, right, towards the column as well as the buffer, right, the molecules will be separate, okay, so they have that particular retention time, and आगे जाके क्या हो जाएगा, buffer gets eluted, and each sample जो मुझे fraction जो मिलता है they can be analyzed, but at the same time जैसे मैंने कहा मैं computerized है this system itself, तो fraction जब जैसे जैसे move करता है they वो automatically अंदर system में ही वो analyze किया जाता है, and on my display, right, मुझे peaks दिखाई देता है, right, because absorption है तो absorption peaks दिखाई देते हैं, and from these, right, sometimes मेरे sample load करने के पहले I can use क्या बोलेंगे standard proteins I can use, right, so that I get peaks, right, peaks मिलता है, and then my sample is loaded, so accordingly क्या हो जाएगा with compared with the standard, right, तो मुझे मालूम गिरेगा कि peaks कहाँ पे आए हैं और वो कौन सा है, कौन सा प्रोटीन है, कौन सा मॉलिक्यूल, is it clear? So this is how, right, HPLC is used, right, for separation of separation and purification. Now separation मैंने कहा तो बड़ा I don't use crude, some amount level of purification already किया जाता है, but HPLC में further purification, जैसे it is used for further purification. So इधर जो sample जो मुझे मिलता है they are in after elution they are in highly purified form, right, purification level has highly increased, okay, so this is what HPLC technique is used for purification. अभी mostly in case in all this अभी अब तक ने जो technique देखा है at the end क्या हो जाएगा, I may get the sample or my enzyme of interest in highly purified form, but sometimes क्या हो जाएगा, they may be in mixture with the solvent, एंड क्या हो जाता है, दिस सॉल्वेंट हैज टू बी सेपरेटेड फ्रॉम द मिक्सचर, तो ताकि मेरा एंजाइम प्यूरिफाइड कॉन्सेंट्रेटेड फॉर्म में मिले। समटाइम कैसा होता है, सॉल्वेंट में रहता है मेरा सैंपल इंटरेस्ट ऑफ एंजाइम ऑफ इंटरेस्ट भी है, बट देन मोमेंट आई से सॉल्वेंट में डिसॉल्व है तो मेरा एंजाइम डाइल्यूशन फॉर्म, तो आई हैव टू कॉन्सेंट्रेट करना पड़ता है। सो लायोफाइलाइजेशन, अल्ट्रा फिल्ट्रेशन, क्रिस्टलाइजेशन आप बोलेंगे, rather than saying purified बोल सकते हैं, बट बेसिकली हम यहाँ पे बोलेंगे दे आर कॉन्सेंट्रेशन टेक्निक्स, राइट। सो लायोफाइलाइजेशन में क्या हो जाता है, यूजुअली माय सैंपल जो है it is in dilute form which having a high level of high amount of solvent I would say. So यहाँ पे मेरा क्या होता है, solvent evaporization करते हैं, but as the name suggests lyophilization क्या है, lyophilization is nothing but a technique, यहाँ पे sublimation type बोल सकते हैं, right, wherein you don't get the liquid, but this all this process is carried out under vacuum, that is main important. So lyophilization किया जाता है under vacuum and the solvent is vaporized and मुझे क्या मिलता है, my enzyme of interest जो है can be obtained in highly purified form. This is lyophilization. Next, another technique which can be employed sometimes is ultrafiltration. अब ये ultrafiltration में क्या किया जाता है, is here I have a membrane, जैसे हम filtration बोलते हैं, use filter paper या something. Here basically ultrafiltration में क्या होता है, membrane is used and membrane के pore size जो होते हैं, they एकदम minute होते हैं, very very minute. तो जब ये माइन्यूट होते हैं, सो यूजुअली अंडर ग्रेविटी ये सेपरेट नहीं हो सकते, मतलब मैंने सैंपल मैंने डाला और बोल दिया वो फिल्ट्रेशन होगा, नहीं हो सकता है, बिकॉज बीइंग पोर साइज बीइंग वेरी वेरी स्मॉल, सो अंडर ग्रेविटी ये सेपरेट हो नहीं सकते हैं। सो व्हाट इज डन इज आई यूज अ प्रेशर, एंड व्हेन आई टॉक अबाउट प्रेशर या नेगेटिव प्रेशर इज एम्प्लॉयड, व्हेन आई से नेगेटिव मतलब क्या, अ वैक्यूम इज क्रिएटेड ऑन द अदर साइड। सो ऑन द अदर साइड वैक्यूम क्रिएट होने के कारण क्या हो जाता है, सेपरेशन हुआ जाता है, राइट। The molecules are pulled, I would say, to the other side through the pores. So जो molecules छोटे रहते हैं pore size से, right, they will move, what say, they will filter out on the to the other side, and जो larger है they get filtered, that means they will remain on the membrane. Is it clear? So this is how ultrafiltration is employed, right, for separation of what to say molecules. And जब मैं vacuum और बोलता हूँ तो sometimes I can use, right, inert gas भी use किया जाता है to obtain the vacuum or negative pressure, such as nitrogen gas or something can be employed, which basically may not employ, say, react with my molecules, but may help in creating a pressure. So this is what is called as ultrafiltration. In all these cases क्या हो जाता है, my sample मुझे मिल जाता है in purified form, and further क्या हो जाता है, I can employ crystallization. Now crystallization technique may not be employed or may not be possible in all, but as the name suggests, crystallization technique is where मुझे purified form तो है ही है, but then I convert it into crystals. Is it clear? Right? Again, now crystals जो किया जाता है, now it depends upon, right? It depends upon either, when I say crystallization doesn't mean I use a high temperature जैसे normal में होता है, नहीं, okay, वहाँ पे तो denature हो जाएंगे, पर मेरा जो sample जो आया है, right, उसको मैं crystallization कर सकता हूँ either by using what you call as sometimes ammonium sulfate जैसा, जो अगर मैं sometimes डाल सकता हूँ, right, जिसके कारण क्या हो जाएगा, my enzyme of interest may get precipitated, and जब precipitate होता है तो they can get converted into crystallized, right, and then filtration करके मैं क्या करता हूँ, right, my enzyme of interest, when I say filtration it can be ultra filtration also, जो भी है, and my enzyme of interest may be in crystallized. Is it clear? So this is how, these are the various techniques employed, all the steps employed to get my enzymes of interest in highly purified. So पहला isolation किया जाता है, isolation होने के बाद में if there is a contamination, so contamination को निकाला जाता है, so I get a purified form, and purified form may be sometimes, right, there may be certain solvents or something जो है, right, उसको further purify या concentration करके, and my enzymes at the end क्या हो जाएगा, my enzymes जो मिले हैं, they will be in a highly purified form, but at the I did not say here, जो starting material मैंने use किया था at the start and at the end अभी जो highly purified enzyme मिलता है, naturally the amount will be very 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 less. My raw material जो use किया था, maybe sometimes it may be in kilogram, and the final product जो आता है purified होता है, तो it may come into a gram or something, and highly purified जो होगा it may be in milligram or maybe it's still less. Okay, right? बट देन जैसे मैंने कहा दिस हाईली प्यूरिफाइड एंजाइम जो है नेचुरली ऑब्टेन्ड, दे मे बी इन they are highly costly and naturally they will be used in certain analytical जहाँ पे लगेगा वहीं पे यूज करेंगे, जहाँ पे मुझे एंजाइम प्यूरिफाइड हाईली प्यूरिफाइड फॉर्म में चाहिए तो वहाँ पे ये एंजाइम यूज करेंगे। अदरवाइज क्या है, आई कैन यूज सर्टेन क्रूड या जो भी है, जैसे हम ट्रीटमेंट जब बोलते हैं तो ट्रीटमेंट में आई कैन यूज क्रूड, मैंने बोला प्लांट यूजफुल है मेडिसिनली, सो आई कैन यूज एंटायर ऐसा ही प्लांट यूज कर सकता हूँ। बट अगर मुझे स्पेसिफिक चाहिए ये मॉलिक्यूल जो काम करता है, राइट, ताकि मेरा उसका एफिकेसी इम्प्रूव हो, राइट, देन वो प्यूरिफाइड फॉर्म में अगर यूज करना है तो नेचुरली मेरा प्यूरिफाइड फॉर्म जो है इट विल बी हाईली कॉस्टलियर रहेगा एंड वो and that molecule which is used in very low dosage because उसका efficacy ज़्यादा है, so it will be used in very low dosage. Is it clear? So this is where, right, I require purified samples. So it depends upon, तो कहीं techniques में क्या है, molecular techniques या जो भी जहाँ पे है, right, biotechnology या जो भी है वहाँ पे मेरे enzymes highly purified form में चाहिए. So this is how the techniques are employed to obtain enzymes in highly purified form. But highly purified form आता है तो naturally क्या है, उसका storage part भी देखना है, because normal condition में वो store नहीं किए जा सकते हैं because they may get denatured. तो उसके shelf life भी आता है. So apart from the cost, उसके shelf life, all these things are involved. Okay, जो भी है, this is how the enzymes are isolated and purified. So with this we complete with the first topic, that is isolation and purification of enzyme. So अभी मैं यहाँ पे 1-2 मिनट का ब्रेक लेता हूँ and I ask you कि अब तक ये जो मैंने डिस्कस किया है, is it everything clear? Any doubt है आपको जो नहीं समझा है? हेलो, अरे अनम्यूट तो हो जाओ और बताओ, या सो गए, छोड़ के गए? हेलो हेलो हेलो,
Students, unmute yourself and let him know whether you have understood or not.
अरे है ना, कितने लोग दिख रहे हैं? सब लोग दिख रहे हैं, फिर आवाज क्यों नहीं कर रहे? बोल की बात क्यों नहीं कर रहे? हेलो हेलो,
Yes, sir.
अरे कुछ बोले, इतना देर लगता है क्या? I know light is sound is slower than light, I can understand, but इतना भी स्लो नहीं है कि उसको मिनट्स में लगे, 5 मिनट लगता है मेरा आवाज आने के लिए। या ऐसा कुछ है कि सर 5 मिनट पहले हमने हाँ बोला था और आपको सुनाई देर से दे रहा है। So any doubt in this अब तक?
Sir, there is a message. Can you share previous slide to have a look?
हाँ, आप लोग है कि नहीं वो देखने के लिए मैंने शेयर निकाल दिया, पार्टिसिपेंट दिखे हैं करके, क्योंकि शेयर साइड में पार्टिसिपेंट है कि नहीं दिखते ही नहीं। शेयर हो गया ना?
Yes.
हाँ,
Yes,
हाँ,
sir.
अभी प्रीवियस स्लाइड मतलब विच प्रीवियस स्लाइड? एक ही तो स्लाइड दिखाया था आज, इससे कौन सा प्रीवियस स्लाइड? यही है ना? हेलो,
Akanksha, which slide do you want? You have to mention.
पहले से बताइए ना, तो कोई डाउट रहेगा तो हम लोग क्लियर कर लेंगे।
मतलब
स्टार्टिंग स्लाइड से, जहाँ से आपने चालू किया था।
आज का या एकदम पूरा?
सर पहले का भी।
अच्छा, पहले का भी? ये देखो,
हाँ।
तो मैंने
it's 11 o'clock, huh?
अच्छा,
Akanksha.
एक मिनट है मैडम।
या और सर यू कैन जस्ट शेयर द स्लाइड्स इन
रुको,
क्लासरूम।
ये मैंने पहले का किया है ना ये सब,
हाँ
ये सब जो है मेरे एंजाइम फर्स्ट पार्ट करके जो है वो डाला था ना।
हाँ सर, वो डाला था आज
हाँ
का भी।
हाँ, तो ये सिर्फ ये नहीं डाला है ना, लास्ट
हाँ
लेक्चर में मैंने ये लिया था, इतना तक तो डाला ही है मैंने।
हाँ, यहाँ तक है, आज का भी।
हाँ, तो इसमें कुछ डाउट्स है क्या? किसको डाउट्स
नहीं,
था?
पहले वाले
नहीं
में तो
नहीं,
कुछ
इसमें
नहीं।
नहीं है।
इसमें कुछ नहीं ना पहले में?
नहीं।
हाँ, ओके। और ये मैंने आज दिखाया, इसको इलैबोरेटली एक्सप्लेन किया ना पूरा।
यस सर।
पिछली बार भी था, पिछली बार हमारा सिर्फ इलेक्ट्रोफोरेसिस तक किया, कुछ हुआ था, आगे यहाँ पे हमने पूरा कंप्लीट किया है, बराबर है? तो एनी डाउट? इसके साथ में कुछ रीडिंग मटेरियल या जो ये मैं डालूंगा ही, इसके साथ में कुछ रीडिंग मटेरियल रहेगा तो वो भी मैं डालूंगा आपको क्लासरूम। ठीक है?
Okay,
Yes,
sir.
sir.
Okay.
Okay.
अभी
Yeah.
next lecture जो जब होगा, then we'll start with the second topic, that is enzyme regulation. Okay.
Okay.
Okay, sir.
ठीक है? तो अभी क्लास टेस्ट जो होगा नेक्स्ट वीक, तो अभी आप ले लो आइसोलेशन एंड प्यूरिफिकेशन ऑफ एंजाइम्स पूरा होगा, ठीक है? तो मैं पीपीटी एज अ पीडीएफ में डालूंगा, साथ में एक्स्ट्रा कुछ रीडिंग मटेरियल होगा वो भी डालूंगा क्लासरूम में, राइट? और साथ में आपको मैंने एक पीडीएफ तो दिया है ना बुक का, वो आपने डाउनलोड
Yes.
किया होगा, तो उसमें से भी
Yes.
पढ़ लो, ठीक है? ओके,
Yes, sir.
सो शैल वी स्टॉप?
Okay.
Yes, sir.
Okay.
ओके, या चलो, थैंक यू, आई विल लीव.
Yeah, so we have 10 minutes break now. Nisha ma'am has already joined. After this, I'll make her the co-host, and at 11:15 sharp, we are going to start. So please join at around 11:13. It's 11:04 now. So sharp 11:15, we are going to start. And I'm giving forward to Nisha ma'am so that she can share. Okay, students, so please join at 11:15.
Hey, recording start.
आणि आवाज क्लियर आहे?
एकदम मस्त.
Okay, okay, okay. Good morning, everybody. Good morning, everybody.
Good morning, ma'am.
Yeah, good morning. How are you all? I hope you all are safe and doing fine. Yeah. Now today we are going to do the second part of the chapter, and that is purine biosynthesis. The last lecture we have completed pyrimidine biosynthesis with all the aspects. The same thing I will be repeating now for purine biosynthesis. Now, students, for this particular chapter, we are going to learn again two types of pathways, that is de novo pathway and salvage pathway, and then later on regulation. Now here in purine biosynthesis, few things once again I want you all to understand, and that is, if you see the next slide here, see you can make out these pictures I've given with reference to these two structures. Now these two structures are purine structures, and these purine structures are two types, and that is adenine and guanine. Now just few seconds, have a look at this. Few seconds, have a look at this. The first one is adenine. What is the nomenclature? That is 6-amino-purine. And the second one is guanine, that is 2-amino-6-oxypurine. I again want you all to just understand the position of these specific groups. In adenine, there is an amino group, and in guanine, there is amino as well as oxygen present. We are going to learn today the biosynthesis of both these, and that is adenine as well as guanine. So please look at the structure once again. Okay.
What do you want to do?
Now this adenine structure which you are looking at with amino group, and the guanine with amino group and oxygen. Now let us go to the next slide. Now this slide shows you, it may appear complicated to you, but it is not. Now this slide shows you the component sources, that is the sources of individual atoms in purine ring. Now here purposely again the same color with the name and the component have been shown. So here look at the first numbering. Numbering will be, look at this, in purine it will be anti-clockwise. So first, second, third, fourth, fifth and sixth. The side ring is 7th, 8th and 9th. The side ring is done clockwise and the main ring is done anti-clockwise. Now here the first nitrogen comes from aspartate, second carbon comes from formyl tetrahydrofolate, then this nitrogen, third and ninth position nitrogen will come from glutamine, the 8th position carbon will come from formyl tetrahydrofolate, then here 7th nitrogen will come from glycine and the 6th carbon will come from CO2. Since the purines are larger molecules, double ring structures, they are going to show you some complications or the prolonged pathway. So here, this is the diagram which is showing the sources of the purine ring. There is another diagram showing you the same structure, and that is okay. So here you can make out the structure in a more simplified manner. So it's the same, but just both the things I found very useful for you all, both the diagrams, and that is why I'm illustrating with them. So here you can understand the sources of these purine components. Okay, students. So what precursors are required for purine biosynthesis are CO2, glycine, then formyl, N10 formyl tetrahydrofolate, then glutamine, again N10 formyl tetrahydrofolate, then aspartate. So these many components are, or these many sources are required to form the purine structure. Now students, one more point I want to tell you here is, in synthesis pathway, biosynthesis pathway, that is de novo pathway, which we are going to learn, which is a major pathway, and it suffices all the requirement of production of adenosine as well as guanosine, or you can say the currency in energy, that is ATP and GTP, is also formed by using these purine rings only. Now in purine biosynthesis, what is a major difference is the rings are attached to the activated PRPP. So activated PRPP should be available first. So it is very different from that of the pyrimidine. In pyrimidine biosynthesis, what we had seen, in pyrimidine, the ring is formed first and then it is attached to the ribose 5-phosphate. So here, the purine ring is formed later, but first PRPP should be available, which are activated, and thus it shows a very peculiar structure. Now here, just to inform, just a few lines to inform that there are two types of pathways leading to nucleotide biosynthesis, that is de novo pathway and the salvage pathway. So de novo synthesis of nucleotide begins with their metabolic precursors, so amino acid, ribosyl 5-phosphate, carbon dioxide, ammonia, and so on. Whereas salvage pathway recycle the free bases and nucleosides released from the nucleic acid breakdown. Both the types of pathways are important in cellular metabolism. Now let us come to this. Okay. Okay. Oh my God. All right. Now here, when you look at this structure, just give me 1 minute. Why this is going so fast? Okay. Huh? This pictures are moving very fast. Okay. Now here we are beginning with the de novo pathway. We are moving with the de novo pathway. Now in this de novo pathway, look at this stage. Now this is the diagram which I am showing you here. This is the typical ribose 5-phosphate. That is a ribose sugar. Ribose 5-phosphate. Okay. This is the 1-phosphate which is attached to the 5th carbon, and this is the typical ribose 5-phosphate. Then when you come to the next, what you can see here is Look at the first step. All right. Now here this ribose 5-phosphate, the first step is phosphorylation. Now this phosphorylation by using ATP molecule, forms AMP. Remember this is the enzyme very well, that is PRPP synthetase. So our PRPP molecule is getting synthesized by phosphorylation. So what happens is, see here, 2 phosphates are added along with the, that is a phosphate groups, 2 phosphate groups are added, and now we get it as the PRPP. Okay, so we are getting that as a PRPP. So now come to the second step. The first step is very easy. First step is very easy. Hey, 1 minute.
स्लाइड में जंप
हाँ,
करने के
ठीक
लिए
है,
ये करना और स्क्रॉल एक स्लाइड को स्क्रॉल करना.
So in the second step, the formed PRPP, now it undergoes amination. The second step, you can see this is going amination. And this amination is with the help of this glutamine. Okay, this is going with the glutamine. Now this glutamine comes in the picture along with the water. It forms PPI, that is inorganic phosphate is released, and glutamate is formed. The enzyme here is PRPP glutamyl amidotransferase. You can make out that on this activated PRPP, the amino group from glutamine is transferred. You can make out here. So this is already the ribose sugar, ribose phosphate, on which now the ring formation has begun. And amino group is transferred. Now when you come to the next step, now students go slow, huh? Try to understand, because there are around 15 to 16 steps which I am going to show you. So here now we go to the third step. Now here in the second step, what is the product? It is 5-phosphoribosyl amine, because it has formed the amino group on the ribose sugar. We name it as 5-phospho. Phosphate is already at the 5th position. So 5-phosphoribosyl amine, amino group is added. And we use the abbreviations like PRA. So abbreviation is PRA. So the first very important component is formed, and that is phosphoribosyl amine, PRA. Then comes the next one. Now when you go to the third step, here what is formed? PRA, phosphoribosyl amine, which is formed in the second step. Now glycine comes in the pathway. Glycine gets added. Once again, phosphorylation of the substrate takes place. The enzyme is GAR synthetase. GAR is glycinamide ribosyl 5-phosphate. So product name is used here. GAR, that is glycinamide ribosyl pyrophosphate synthetase. Now what has happened here? Here the glycine donates this complete portion. Okay, so this is now the formation of the ring in progress. So here the glycine donates this COCH2 as well as NH2. So glycine also contributes mainly for the formation of purine ring. So this is the third step. Now when you go to the fourth step. When you go to the fourth step, look at this. Fourth step. Okay. So I hope you have seen this third step properly, and this is the product glycinamide ribosyl 5-phosphate or glycinamide ribotide, that is GR. Okay. Now come to the next one. Okay, here what you can see here is, all right. Okay. Now fourth step. Now today this, okay. So glycinamide ribotide, that is a GAR, was formed earlier. So now what happens is, see, the next step is that N10 formyl THF come in the picture. So it is formyl tetrahydrofolate comes in the picture. The enzyme is GAR formyl transferase. So formyl group is now transferred. Okay, so formyl group is transferred, and you can see here I don't know, today my slides are not giving me proper.
Do you want me to show the cursor?
No, I have, okay, it's okay. So this tetrahydrofolate is released, and what is formed is now formyl glycinamide ribotide. Here, the formyl glycinamide ribotide, and that is FGAR. Okay, so this is the next step come in the picture. Okay, now just try to, just students, just fixed. Okay. Okay. So fourth step, as I said, GAR formyl transferase. So here, this formyl particular, this continuous here, you can make out this structure is added here. And now, see students, try to understand one thing. This ring formation contributes to the side ring, secondary ring of the purine, which with 5 atoms. This is not the main ring. So first, in for your convenience, if I say right side ka ring. So here this ring formation is taking place, and now you will find that here the compound is formyl glycinamide ribotide. So FGAR. This is FGAR is formed. Okay, are you getting that? So here the ring formation here is with almost completed, and it is now attached with the ribose phosphate. It is already attached with the ribose phosphate. So ribose phosphate holds the entire structure step by step and shows the formation of ring structure. Now if you go to the next, and that is this formyl glycinamide ribotide FGAR. Now we are studying step 5. Once again, that glutamine comes in the picture. It gives its component forming the glutamate, and here you can make out that the structure is now little bit changed. This is the formation of the side ring, and the compound which is formed is formyl glycinamide ribotide FGAM. We'll go to the next step. I'm going to show you the complete overall pathway also in a simpler manner. Now, if you come to the sixth one, that is formyl glycinamide ribotide, our earlier precursor or substrate. Now it undergoes phosphorylation, and this phosphorylation shows utilization of ATP, removal of water. It is the enzyme is AIR synthetase, air synthetase. What does it mean? Air synthetase means amino imidazole ribotide. If you see this, the structure here, this is amino imidazole ribotide. So amino group is added. This ring is imidazole ring, and ribotide or ribose is attached, and therefore it is air, AIR, and that is amino imidazole ribotide. Okay, so this is the step. Now we will go to the next, which is 7th. Now here if you look at the 7th step, it is aminoimidazole ribotide AIR. Now the enzyme is AIR carboxylase. Carboxylation takes place. When the carboxylation takes place, here you can make out you can make out here that the carboxylation takes place and the major component is formed. You can visualize here. You can see the formation of the double ring started. The second the primary ring formation is started. So CO2. So carboxylation has begun with the formation of major ring, and the side ring with CNCHN. The nitrogen at 7th and 9th position is already there. Amino group is there, and now you can make out the actual proper purine ring is getting formed. Now we come to the 8th stage. 8th stage tells you or shows you this CAIR or carboxyaminoamidazole. This carboxyaminoamidazole is now getting involved with aspartate. The third amino acid comes in the picture, and that is aspartate with ATP. Now it forms a major structure of the ring towards the completion, and now it forms aminoimidazole succinyl carboxamide ribotide, or sometimes it is also known as succinyl aminoimidazole carboxamide ribotide. So it is SICAR. We call this as a SICAR, and the enzyme is SICAR synthetase. I repeat this step because this step is very important for the formation of second or that is a major ring. So carboxyaminoimidazole ribotide CAIR undergoes the reaction with aspartate plus ATP. The catalyzing enzyme is SICAR synthetase, and now it forms in the process the molecule of this SICAR or succinyl aminoimidazole carboxamide ribotide. Now come to the 9th stage. 9th stage, the fumarate molecule goes out, leaving only COO, leaving only the COO of this. See here, leaving only this C double bond O, everything goes out of the cycle, forming the forming the fumarate. So fumarate goes out of the cycle, and now the proper component is formed of the major ring. So here NH2, C double bond O, and NH. The compound is aminoimidazole carboxamide ribotide, AICAR, or you can say ICAR. Now this ICAR, when we consider for the next step, you can make out here that this particular compound, when it goes for the next process, N10th formal derivative of tetrahydrofolate comes in the picture, then donates this formal molecule with the help of catalyst enzyme, and that is AICAR formal transferase, because formal derivative is transferred. And here you can make out the component which is formed as formaminoimidazole carboxamide ribotide, that is FAICAR, which is still attached, of course remains attached with the ribose 5-phosphate. The next step is 11th number. For convenience, we have numbered it. So this FAICAR now gets converted into the next product by release of water molecule. The water molecule 2HNO, H2O is removed, and now students, you can see here the complete ring of purine is formed. Now this major ring is completely formed, and it is attached to the ribose sugar at its first carbon. So if you check here very properly, on the ribose 5-phosphate molecule, which is this one, at the first carbon, entire double ring structure of purine is placed. Now this whole structure, that is the complete activated ribose sugar and completely synthesized purine ring, when you observe or when you get, it is the key component of the de novo pathway, and that is inosine monophosphate. This
मैम
is hello.
मैम स्लाइड चेंज नहीं हो रहा है, स्टेप 4 का हम लोग को स्लाइड दिख रहा है.
अच्छा, स्टिल स्लाइड 4 ही दिख रहा है, सब लोगों को स्लाइड 4 दिख रहा
यस
है.
मैम.
Yes, ma'am.
Oh, and I have reached already. You should have told me earlier, dear.
मैम हम लोग ने चैट बॉक्स पे डाले.
अच्छा, okay. रुक, I'll do it again. 1 minute, हाँ. That's enough. कि slide 4 के बाद दिखाई नहीं कुछ भी. Slide change ही नहीं हुआ. Just hold on, I will repeat again, okay?
Can you see now?
अभी दिख रहा है क्या? Now just can you see? I'm moving
Yes,
the slide.
Yes,
Okay. Okay, so I'll do again. Okay, I'll do it again.
Yes, ma'am.
हाँ.
This now.
Okay. Now shall I just quick, this is okay now, this 4th slide I had shown, 4th step.
Yes, ma'am.
Yeah, okay. So glyceramine ribotide and then N10 formyl THF comes in the picture. Okay. Now the next one is formation of formyl glycinamide ribotide, right? Formyl glycinamide ribotide. Then, okay, I'll do it from 5th slide once again for you all. So formyl glycinamide ribotide now shows the reaction or entry of glutamine along with the ATP molecule. Glutamate comes out, ADP is formed. The enzyme is formyl glycinamide. This is formyl glycinamide. Look at this. I'll just show you the structure. So formyl glycinamide amidin. Now it forms a formyl glycinamide ribotide, and that is FGM. Here what I said is, here you can make out the ring formation is almost complete. The ring formation of the purine. But what I said, this is the first side ring. It is not the major side ring. This is the first one towards the right hand side wala ring. Secondary ring is getting formed. Now when you come to the next slide or when you come to the next step. So this formyl glycinamide ribotide undergoes phosphorylation. The enzyme is AIR synthesis because the molecule which is formed here is amino imidazole ribotide. So side ring is known as the imidazole ring and it forms amino imidazole ribotide, that is AIR. When you go to the next, which is more important, which you have to know. Now here, this amino imidazole ribotide, that is AIR, undergoes carboxylation. So AIR undergoes carboxylation. So obviously the enzyme is AIR carboxylase. When you look at this, see COO, that carbon dioxide is incorporated to form now the first ring, which is very primary ring. So here the secondary imidazole ring is completed and the primary ring is getting formed. So the compound here is carboxy amino imidazole ribotide. So carboxy, because carbon dioxide is added, so carboxy amino imidazole ribotide. Now when you go to the next step, aspartate comes in the picture, it gets phosphorylated, and now see, this aspartate donates the major again gets incorporated and it is going to help to contribute this NH for the formation of further ring. Here cycar synthetase is involved and the compound is amino imidazole succinyl carboxamide ribotide. It is also known as succinyl amino imidazole carboxamide ribotide. So cycar. When you come to the next step, that is a fumarate. This entire derivative which was added, entire portion which was added from aspartate only NH2 is retained, the fumarate goes out and the ring is formed. Now the completion of the ring is only one step behind. And in the next step, you will see this aminoimidazole carboxamide ribotide at the 10th step. It shows involvement of N10 formyl THF, tetrahydrofolate. The tetrahydrofolate comes back and the formyl derivative is added into the ring, and that is here. That is the CHO. And now in the next step, okay, this compound is amino-imidazole carboxamide ribotide, and that is FAICR. And at the 11th step, water is given out and the formation of ring is completed. Here the formation of ring is completed. The double ring purine molecule is completely done on the first carbon atom of ribose sugar. And here you can make out that this ring is the primary purine ring. Now, according to the further requirement of synthesis of ATP or GTP or AMP and GMP, the further minor changes will be occurring. But now the major precursor here or the intermediate here is inosine monophosphate, that is IMP. You can remember from the abbreviation, this is very important. This particular substance or product is very, very important, and that is inosine monophosphate or IMP. Just have a look at the structure properly, and that is, this is the basic ribose sugar. At the first carbon, this basic purine rings are constructed. So what we had in all the steps to be noted is activated PRPP formation first, then this ring, imidazole ring formation, and then followed by the major ring. And here you can see two ringed structure, that is a basic purine structure is formed. And now the things which you are going to learn are the actual steps where AMP and GMP are formed. Here I will be giving you some key points by which you can remember the bifurcation process. So in the next step, what I have shown here is, this is the inosine monophosphate, our major intermediate. It is going to now show bifurcation towards the formation of AMP and GMP. Now let us see this step. Here, for formation of AMP, remember students, aspartate comes once again in the picture. The energy source is GTP. This point I want you all to note very well, that for AMP, the amino acid comes as aspartate, but the energy currency is GTP. GTP gets broken down to GDP, the energy is released, and the next step occurs or results in the formation of adenyl succinate. This is adenyl succinate synthetase. Aspartate gives the entire this component. The total entire this component is donated by aspartate. And here the formation of adenyl succinate takes place. The next step here, this adenyl succinate gives away the fumarate molecule. Fumarate, this is fumarate molecule. The enzyme is adenyl succinate lyase. And here you can make out the formation of adenosine monophosphate. Amino group is added. From aspartate, only amino group is retained. Rest of the substance or rest of the atoms come out as a fumarate molecule, and AMP is formed, which is attached to the ribose 5-phosphate, and this is the complete nucleoside along with the nitrogen base and the ribose sugar. So here is the AMP formation. Why did I tell you here to remember that formation of AMP, the energy currency is GTP. It is the reciprocal balance, student. It is a coordinated reciprocal balance for energy currency molecules. Now come back to the next diversion here, the another diversion. This IMP molecule now undergoes simple, that water enters and a reduction process, and that is NADH is formed, and enzyme is IMP dehydrogenase. So hydrogen is removed, NAD gets reduced to NADH, forming one free proton. And here now you can see here the oxygen is added here, and the compound which is formed is xanthosine monophosphate. It is attached to the ribose 5-phosphate, and this is not yet completely GMP, but earlier precursor is xanthosine monophosphate, the early precursor to GMP. Now to this xanthosine monophosphate, glutamine is added. Here this glutamine is added and amino group. Instead of this oxygen, we are going to get now the amino group, and here you can make out amino group is added, oxygen is retained, and this is now guanosine monophosphate. So this is guanine with ribose, so guanosine monophosphate. For formation of guanosine monophosphate, energy currency is ATP molecule, and the enzyme is transamidinase, because the amino group is transferred to the ring, and therefore it is transamidinase. So here we learned about the formation of AMP and GMP. Now students, the entire de novo pathway of the purine is a very long process, involves almost around 15 steps, so many enzymes and many sources, amino acids, carbon dioxide, ATP, GTP molecules, and so on, activated PRPP, on which these double ring purine structures are formed. But if you go step by step and notice the changes, it will not be difficult for you. Now, as I had said, that I will be showing you the proper pathway once again in a abbreviated form. This slide I will be not discussing right now, but here just have a look at this. Okay, this is I have prepared for you. This structure or this particular write-up I have prepared for you, and that will show you the various steps. Let us revise quickly. So we are learning about the de novo pathway for purines. So first molecule, I'll just go quick, like a overview. So it's a ribose 5-phosphate. That is a D-ribose 5-phosphate, अपना regular ribose sugar. It undergoes phosphorylation with the help of PRPP synthetase. It's a key enzyme for regulation also. So here phosphoribosyl pyrophosphate is formed. In the second step, glutamine enters. It forms a glutamate. Water enters also. It gives away this pyrophosphate group, and it forms 5-phosphoribosyl amine. Amino group is added from glutamine. Now this once again undergoes amination, and that glycine is involved in the reaction with the help of enzyme GAR synthetase, because glycinamide ribosyl pyrophosphate is formed. GAR. So GAR synthetase. The next step which you come across is the fourth one. N10 formyl THF tetrahydrofolate comes in the picture. This formyl group is added, and therefore it is formyl glycinamide ribotide. In the next step, this glutamine is involved. It gives glutamate. Glutamine will give one more amino group, and it forms formyl glycinamidin ribotide. It is a formyl glycinamide ribotide. In the next step, that is the sixth one, phosphorylation takes place, and this formyl glycinamide gets converted into amino imidazole ribotide. So side ring synthesis is done. At this step, the side ring synthesis is done by using the ATP molecule. The next step, when you come, carboxylation takes place, and therefore carboxy amino imidazole ribotide, main ring structure, starts forming at this state, and the compound we name it as CAIR. After this step, when aspartate comes back in the picture, and it forms now amino imidazole succinyl carboxamide ribotide, and that is SAICAR. This step further occurs where the fumarate comes out, And this fumarate molecule is given out with the help of adenylosuccinylase, and the compound form is aminoimidazole carboxamide ribotide. Here, the next step, 10th one, formal THF comes in the picture and forms now form aminoimidazole carboxamide ribotide, that is FAICAR. Water comes out in the next picture, next step, water comes out, it's a cyclohydrolase, and now the compound form is inosine monophosphate. Primary purine structure is totally done. So here I have shown you the purine base ring, which is the primary purine ring is formed and it is attached to the sugar molecule. Now here, bifurcation, I have written down for you all almost in the same manner in which I've shown you the slides earlier. And I wanted you to understand certain minor points. So aspartate for AMP, but GTP for AMP. And here, when this aspartate is involved, with the help of enzyme adenyl succinate synthetase, this entire component is added. Purine ring is almost formed. And here, when this purine ring is formed, the fumarate derivative is removed. This entire fumarate derivative is removed. Amino group is placed, and what you get is adenosine monophosphate. When you look at this adenosine monophosphate, you know the chemical nomenclature, it is 6-aminopurine. This is 6-aminopurine. This AMP is done. AMP is, this is adenine and attached to the ribose 5-phosphate. Similarly, when you come to the second half, okay, here this IMP or inosine monophosphate is now when it is involved to form the GMP. So here the IMP dehydrogenase enzyme removes the hydrogen. Due to that, NAD gets reduced to NADH, 3 protons are formed, and here the molecule which is known as a xanthosine monophosphate takes place. Formation of xanthosine monophosphate takes place. Now further to this, the glutamine is added. Again, you can remember GMP, so glutamine at the last step. Glutamine is added, phosphorylation by ATP. Energy currency is ATP. Glutamate is formed and this amino group is added. Here, this amino group is added and guanosine monophosphate, we call it as a 2-amino and 6-oxy. What was the AMP? Adenine was 6-aminopurine, whereas here 2-amino 6-oxypurine. These changes takes place only when IMP comes in the process. So here, students, you can see that the GMP and AMP both are formed as a end result of occurrence of de novo biosynthesis pathway. Now, students, today I'm going to take only these. I decided to take only this pathway for you all because there are so many reactions. There are so many reactions, and it is not very, very easy at the first instant to understand that and to digest. So as far as the explanation is concerned, I will stop here. And now you all can talk to me if it is okay for you. Learning this biosynthesis pathways was okay for you. You can unmute yourself and let me know.
Ma'am.
Hello.
Yes, ma'am.
See, this entire purine biosynthesis pathway I will be uploading today itself, and you can see that. Okay, you can see that very clearly. Okay, now if 1 minute, let the slide be on the screen. So anything you want me to explain once again? Basically, students understand this. See, 15 steps biosynthetic pathway is going to take you time to follow, to understand. What I would suggest is when you study this pathway, look at the structural changes. The structural changes are very, very gradual, and then you will be able to remember this complete pathway easily. Okay, okay. Somebody was saying something.
Ma'am, Pankaj has left a message that due to power fluctuations, he is not going to attend. I mean, he won't be able to attend.
Okay, okay, okay. Anybody who are present here have a doubt?
Ma'am, can you hear me?
Yeah, I can.
Ma'am, I just want to ask, during from inosine monophosphate, the
Yeah.
bifurcation to adenosine monophosphate and guanosine. Ma'am, while discussing about pyrimidine biosynthesis, you told that only when DNA is required,
Ha.
that time
Okay.
thymine is made. But ma'am,
Ha.
what
Ha.
during
Correct,
this,
correct, correct. Yeah.
madam,
Yeah.
यहाँ
Now this doubt I correct, correct. Now this doubt I will tell you because this is a part of the next lecture. Listen. Now here, what I showed you, just a simple basic nucleoside structure, and that is with the ribose sugar, right? Okay,
yes, ma'am.
with the ribose sugar. Now you must be thinking that AMP, GMP are formed. So these are the basic nucleosides, of course, with the reference to ribose sugar. So they are directly going to be the component of RNA. Am I right? So
Yes, ma'am.
when they are going to be the component of DNA, because adenine and guanine are present or are the component of DNA molecule also. So
Yes.
now it's a very, very logical point which you have raised, and I would answer in this way is just simple one step goes ahead, which I had explained you in the last lecture, that ribose sugar gets converted into deoxyribose sugar.
Yes, ma'am.
Do you remember that? Ribose sugar, when it gets reduced to deoxyribose sugar, the entire nucleoside which is going to be formed is DAMP or DGMP. Am I right? Are you
Yes,
getting
ma'am.
that?
Yes,
Hmm. So here the adenosine monophosphate is the basic structure which are formed by de novo pathway. If they have to be incorporated in DNA molecule, and that is only required during DNA replication, correct? So DNA nucleotides are required only during
yes, ma'am.
DNA replication. So during DNA replication, when it is required, the ribose sugar gets converted to the deoxyribose sugar by which enzyme? Do you remember that? Reductase, ribotide reductase. Clear? So that riboside
Yes, ma'am. Yes, ma'am.
reductase enzyme, last time I had explained you that. Okay, if you go to the
Yes,
PDF and
ma'am.
if you see that, I had given you the special illustration that how ribose sugar is reduced to deoxyribose sugar. So here, the
Yes,
same way, AMP gets converted into DNA AMP or DAMP or DADP. Are you getting that? And
yes, ma'am.
hence, the nucleotides which are required for DNA replication, they get attached to the deoxyribose sugar because now ribose sugar is reduced to deoxyribose. So automatically they become DNA nucleotides. Is it okay? Is it clear to you?
Yes, yes, ma'am.
So this,
Ma'am, one more question.
bol.
Ma'am, my question was when this will decided from inosine, I will that pathway is going to make adenosine and
Yeah,
other
yeah,
pathway is going to make
correct.
guanosine.
So now what happens is, see, two things you have to remember. Formation of pyrimidine and purine are always in coordination because the total number of purine is equal to total number of pyrimidine in DNA molecule, whereas it is not the
Yes, ma'am.
same in RNA molecule. So it depends upon for which the nucleotides are required. Okay. Now, if the nucleotides are required for DNA molecule, both AMP as well as GMP has to be formed. Adenosine is also,
Yes, ma'am.
adenine nucleotide is required as well as guanosine nucleotide is required. So that time, this both the pathways go simultaneously. Whereas in RNA transcription, as per the requirement, because RNA transcription takes place as per the DNA strand which is getting transcripted. So as
Yes,
per
ma'am.
the requirement, the regulation takes place. That regulation I'm going to take in the next lecture. That how it is decided that whether adenine has to be formed or guanine has to be formed, or out of the three pyrimidines in pyrimidine regulation, I've already told you that how it is
Yes, ma'am.
controlled. So when I take the regulation of de novo biosynthesis pathway in detail, That time I will tell you how it is decided whether to form adenine or guanine, but basically as per the requirement, as per the cellulose requirement. If suppose GMP is not required, then the step which I have shown you here, if the GMP is not required, then here this IMP dehydrogenase enzyme is there. Na? Can you see this? So this IMP dehydrogenase enzyme is under the regulation. No dehydrogenation, and automatically this step is controlled. Only aspartate will come in the picture and no glutamine will come in the picture. Are you getting that? So in the regulation, I will be telling you how it is decided, which purine is to be formed, which purine has to be controlled, and how the entire de novo synthesis is regulated. Is it okay?
Okay.
Yeah. Anybody else?
Thank you, ma'am.
Yeah. Now, anybody else to have a doubt or have a question? Yeah. Unmute yourself and let me know. Okay. Everybody has to talk. Sadaf, are you there?
Yes, ma'am.
Any doubt? You must ask certain questions. Okay. What I will think is first learn this pathway, and in the next lecture, whenever it is arranged, I will take the revision of this pathway plus regulation of purine biosynthesis plus salvage pathway, right? And I will be telling you few points which will help you to understand what is the significance or what are the effects of this de novo pathway of purine and pyrimidine also.
Yes, ma'am.
Yeah. Okay. So shall I stop here now?
Okay.
Okay. Okay, Veena, is it okay now? Timing is done?
Ekdam mast. Perfect.
Barabar. Okay. Okay.
If you stop the screen, share screen, then
Okay. Stop. I will stop the share screen.
yes, yes.
Yeah.
Thank you.
Yeah. Okay. Thank you.
Bye, take
Thank you so
care.
much. Yeah. Bye. Bye. Take care.